Hi, everyone. Welcome to this episode of Crops TV. Thank you for joining us. We are going to be talking about potential strategies to mitigate yield drag in corn following a winter cereal rye cover crop. I'm Alison Robertson. I'm a professor in plant pathology, entomology, and microbiology, and I'm also an extension field crops pathologist here at Iowa State. I've been working on cover crops for about 12 years now. And with me today, I have some colleagues who have been helping me with this research. And so I'm going to ask them to introduce themselves. Peter, do you want to go first?
Yeah. Hi there. I'm Peter O'Brien. I'm a research agronomist with the United States Department of Agriculture, Agricultural Research Service.
Hi, I'm Rochelle Matheson. I'm a research scientist at Iowa State. I work in Dr. Allison Robertson's lab on various things of cover crops and seedling disease.
So I'm going to give a little bit of a background information to some of the research that we've done. And then what we're going to do today is we're going to share research that Rochelle's been doing and that Peter's been doing along with us. So first of all, I just wanted to talk about the background to this research. So what we're predominantly interested in is corn yield drag following a winter cereal rye cover crop. And this is some data from the Iowa Learning Farms, probably getting a little bit old now, but just showing that sometimes when you plant corn following a winter cereal rye cover crop, that there can be a reduction in yield. This doesn't happen in every field. It doesn't happen in every year. And so what we're trying to do is understand why this happens. So a lot of times what we find is that with corn planted after a winter cereal rye cover crop, we may have an uneven stand. And that uneven stand can be the result of many different factors. And so all of those factors have been hypothesized to be responsible for this yield drag. And those factors include uneven planting depth, low soil temperatures, allelopathy, and then also nitrogen immobilization. What we showed probably about 10 years ago is that winter cereal rye is a green bridge for corn seedling pathogens. So what do I mean by that? When we plant cereal rye into the soil, there is a microbiome that is associated with that cereal rye. And what we found in our research is that when we terminate the winter cereal rye, we actually get an increase in the number of seedling pathogens that you see once that rye is terminated. Eventually, if those seedling pathogens don't have a host to infect, they will decrease back to their background numbers. But if we plant corn while those numbers of seedling pathogens are very high, then we run into a greater risk of seedling disease. So when we think about plant disease management, there are 6 traditional principles that we talk about. And what we're going to be focusing on today is exclusion and avoidance. So this means that planting when or where the inoculum is low or not present. So you can see in the cartoon below, this shows you how those pathogens increase when the winter cereal rye plant is terminated and then decrease back to background levels. And so what we would do is we would wait to plant the corn, and therefore by waiting to plant the corn, we're allowing those seedling pathogen populations just to decrease back down to those background levels. And so one of our recommendations is to plant corn at least 10 days after terminating that cereal rye. Now, there's a lot of interest in planting green. Of course, when we plant green, then we're planting when those population numbers are much higher. The other thing that we could do is we could plant where the inoculum is low or not present. So this is something like planting a skip, leaving a skip row, right? When you drill that cereal rye and then planting that corn in that skip row, it's going to be further away from that cereal rye. And so in theory, there should be less pathogens there. So what we're going to start with is just talking about some research that Rochelle was doing, which looks at this aspect of planting when the inoculum is low. So as I mentioned, we'd rather wait at least 10 days until after the cereal rye is terminated, and then plant the corn. But Rochelle has been doing a multi-state project where we've been looking at the effect of planting green. So what happens when we plant that corn at rye termination when those population levels are high? So Rochelle, would you like to tell the listeners about your research?
Sure. Thanks, Allison. So we were part of a multi-state project with the Precision Sustainable Agriculture Group. It was funded by the USDA. And in Iowa and different locations, we were part of what was called Common Experiment 2, where we evaluated the effect of a winter cereal rye termination timing on various pests, such as slugs, different insect pests, diseases, weeds, and then looked at corn development and performance. So the locations that were participating in the Common Experiment 2, you can see here across the United States, there were 16 different locations. And the co-PIs of this project was Stephen Mirsky from the USDA and Chris Ryberg Horton from North Carolina State. And we had plant pathologists, entomologists, and weed scientists at the 16 different locations that made up this group. Allison here was for the plant pathologists, and John Tucker at Penn State was the lead for the entomologist, and Dr. Carlene Chase was the lead for the weed scientists. And so we all worked together doing a bunch of different experiments for insects, weeds, and diseases. So for this project, we had different treatments where the winter cereal rye was terminated at different times before the corn was planted. And we had 3 treatments one was called planting brown, where the cover crop was terminated using glyphosate, and that was at 14 or more days before corn planting. We also had a planting brown green treatment, which was the cover crop was terminated 3 to 7 days before corn planting. And then finally, we had planting green, where the cover crop was terminated at planting or up to 7 days after corn planting. And then we also had a bare soil treatment where the rye was not planted. So going into looking at the different factors that we looked at for this project, I'm just going to be talking about those related to seedling disease. So we will be publishing this work with the insects and weeds and that kind of thing later. So I'm just going to focus on the winter cereal rye biomass, the seedling disease, and the corn yield for now. So looking at the biomass, not surprisingly, across the locations and the 3 years, the greatest amount of biomass that was produced was in the planting green treatment. And you can see there for the 3 other treatments, there was quite a range across locations and years of how much biomass was produced. But the take-home message is the greatest amount of biomass was in the planting green treatment. For the corn seedling disease, everyone collected seedlings, corn seedlings, and we then assessed those seedlings for root rot severity. And what you can see here across the different years and locations is that there was a range of root rot severity across the treatments. But the kind of take-home message again is that we saw that root rot was most severe in the planting green treatments for the majority of locations and years. And looking at another thing we looked at was corn yield. So we looked at relative corn yield, and for the figure that's here, what we did is we looked at planting green compared to planting brown specifically. So what we found is, again, there was a wide range of relative corn yield across the locations and the years. However, one trend was that relative yield was negative at the majority of locations for all of the years. And that just means that planting green had lower yield than planting brown. Another thing we looked at was the relationship between different factors. And so for here, we're looking at the winter cereal rye biomass relationship with corn root rot severity. And so for this figure, the top row is all termination timing treatments, whereas the bottom row is planting brown and planting green only. And what we found was that there was, first off, a positive relationship for all of the years, and that just means that as one factor increases, the other factor increases. So for here, it's looking at winter cereal rye biomass that increases, and then root rot increased. And also for 2021, which was the first column, we saw a moderate positive relationship. And then for 2022 and 2023, they were weak relationships. And that just means that the strength between those two factors was weak to moderate across the 3 years. So for the other two factors, we looked at corn root rot severity and corn yield. Again, the top row is all termination timing treatments, and the bottom row is planting brown and planting green. And what we found for these is that in 2022 and 2023, there was a negative relationship, meaning that as one factor increases, the other factor decreases. So for what we're talking about here is as root rot increased, then yield decreases. However, in 2021, we did not see a relationship between those factors. As you can see, the line is, it doesn't deviate. So in 2022 and 2023, then we saw that there were a weak relationship between those two factors. Okay, to summarize this project, what we found was that planting into a winter cereal rye cover crop may negatively affect corn performance. So that can be due to delayed emergence, increased seedling disease, or yield drag, especially if producers are thinking of planting into a green cover crop. So are there any questions about that?
Yes, I have a question, Rochelle. So it looked like, looking at the data, that with some states there seems to be a problem with seedling disease, with other states not. Sometimes there was a difference between years in the amount of seedling disease and whether there was a yield drag. So I was wondering, are you planning to do any further analysis of the data to look at that perhaps?
Yes. So what we're planning on doing is looking at all the different factors that we can think of, such as precipitation, highs, temperatures, low temperatures, different factors, also including the soil type and those type of factors to then see if that had an impact or that we can explain why we were seeing differences between years and across locations. And so we will be doing that specifically with seedling disease or the plant pathology part of this project, but we will also be doing that collaboratively with the other disciplines, so with the weeds and the insects also, and kind of throwing everything together to see if we can sort of start explaining why we're seeing yield drag for multiple different factors.
I think the most exciting thing for me as a plant pathologist, maybe not for a farmer, is that this work that we did, we already knew in Iowa that planting green, we would see increased seedling disease and perhaps yield drag. And so the exciting thing for me with this project was that it's not just Iowa, that it does happen across the country, and therefore it's something to do with the production system. And if we can understand what it is, then perhaps we can manage it and mitigate that yield drag.
And help farmers understand.
Yes, yeah.
Okay, so I think now we're going to be talking about inoculum in the soil. Right?
Yes.
So I'll send it back to you.
I'm going to be talking about some research that a master's student did with me, Sarah, a couple of years ago. But this is where we're trying to plant the corn where the inoculation is low. And when I first presented this work, I had a farmer say to me, "Oh, you're social distancing the corn and the cereal rye." And so we fondly call this work social distancing, because that's exactly what it is. So in Sarah's work, what she did was she had these small plot trials where she socially distanced the corn from the winter cereal rye, right? So we always have a no rye control. And then we have the rye that is drilled down the bottom. So that now we're planting the corn directly into the rye. So for, in a lot of cases, that corn seedling is no inches from the rye, right? 0 to about 3.5 inches from that rye. And then what we did when we drilled the rye, we blocked one of the drills over the corn row. And so we were able to plant 3 rows of rye in between the 30-inch corn rows. And so that meant the corn was now 7.5 inches away from the rye. And then we also plugged 3 of the drills, and so we seeded that winter cereal rye on 30-inch rows immediately in between the corn. And what Sarah did here was she looked at biomass, seedling disease, and yield. You can see that in the 2 years we had a difference in the amount of biomass that was produced. Obviously, we had the most biomass where in the 0 inches away from the corn, and that decreased as we went down to that 1 row of every 30 inches of the winter cereal rye. If we look at the seedling disease, so you can see the treatments here, you can see the percent of the radicle that was rotted on the left-hand side, those figures. And what you'll notice is that we had the most rot in that drilled corn, more than half the rot in our no rye treatment, but we actually had less rot in the corn that was 15 inches away. And this was for 2020. So in 2019, we had very low disease severity, and we didn't see any differences between the treatments. And so, yes, just by socially distancing that corn from the rye, it looked like we could reduce disease under high disease severity years. And then when we look at the yield, the slides laid out the same way, but the yield in 2019 and 2020, you can see that in 2019 that we had the greatest yield in our no rye control, and where we socially distanced that corn 15 inches away from that rye, we had very similar yield. When that corn was planted closer to the rye or in the rye, those yields were quite a bit reduced. In 2020, we have the greatest yield with the no rye, but where we separate that corn 15 inches from the rye, we had about a 10% yield reduction compared to a 50% yield reduction where we planted the corn into the drilled rye. So this was really exciting research, and it showed us that socially distancing the corn from the winter cereal rye cover crop could be advantageous in that we would get better growth of that corn, we could reduce the amount of seedling disease, and we could also mitigate the yield drag that sometimes we see when corn is planted following cereal rye. But if you think about why we're planting a cover crop, there's some shortfalls, right, with social distancing the winter cereal rye from the corn. So when we look at if we have no rye on the landscape, then we have 0% cover. If we drill that rye onto the landscape, then 100% of our field is covered with residue. And when we socially distance the rye from corn, we now have 25% of the land covered with cereal rye, or 75% less. And so that brings up a lot of questions, right, regarding why are we planting this cereal rye? We're planting it for ecological and environmental reasons. And Peter, do you want to just address why, some of the issues that we could get into with planting less rye on the landscape?
Yeah, absolutely. So farmers use rye as a cover crop for a lot of different reasons. And in Iowa, two of the really most important ones are erosion control and to reduce nitrate leaching out the tile drainage. And really both of those benefits depend on maximizing cover crop coverage and biomass. And so even just looking at this slide, you can tell that when you plant the rye in less frequently or fewer rows, you're just getting less soil surface coverage. And so you're protecting less of that soil surface, especially before the rye has much time to grow, which is when it's most susceptible to erosion. And so altering our cover crop management brings up a lot of questions about how much can we alter it and still maintain those benefits. And those benefits that I mentioned are definitely, you can see in the center there, a risk of a field without any green coverage early in the spring, a lot of water coming off, a lot of soil going with it. But there's also other cover crop benefits, thinking about soil structure, thinking about weed suppression. All of these things kind of play into where the rye is on the landscape and how much of it there is on the landscape. And so kind of a natural progression of the work that Rochelle and Allison have described already is, what if we combine those two strategies of planting green or separating the rye from the corn in time, and social distancing, where we separate the rye from the corn in space. And so Allison, do you want to describe an experiment we set up to explore that?
So, this experiment was such a nice progression from the work that we'd already done, and Peter was gracious enough to join us and help with the erosion and etc. part of the study. But so what we did was we had our positive control, if you like, or negative control, a control, where we planted the corn into drilled rye. And but in this case, we had the rye that was terminated early, so at least 7 days before planting. And then what we did was we planted the corn into the green rye, and so we terminated that rye at or soon after planting. So now we have the separation in time and the separation in space. We needed to combine that. So the next thing we did was the skip row, where we're planting those 3 rows of rye in between the corn. Once again, we have the planting green and the planting brown, to use Rochelle's terminology there. And then we did the 30-inch, where we planted the rye in 30-inch rows, so 15 inches away from the corn. And we did the planting brown and then also the planting green treatment. And of course, in our cover crop studies, we always include a check of the no winter cereal rye. That's what our goal is, to try and get corn growing as well as it does after no winter cereal rye. So that check is always there in the background. So Peter, do you want to discuss what we found in terms of the rye biomass? Because that was really fun over the 2 years that we did this study.
Yeah, absolutely. And it was, as all these studies tend to be, the 2 years were very different from one another. And so we have a picture showing that on the left, where that was from 2023 in the spring, which I would call, roughly an average amount of biomass production, probably a little bit below average. But then on the right there, you see from 2024, a massive, massive amount of biomass. And this is almost what I think farmers would be concerned about with putting cover crops on the field, is what if it gets away from me this way, and I have rye biomass that's taller than I am when I walk through the field. So those are kind of our two conditions that we have to deal with, that we're going to talk about. And so basically, in 2023 especially, it really followed the trend that we'd expect from if we're planting less rye on on the landscape in the skip row and the 30-inch row, we're getting less biomass. And then within those, if we let it grow a couple weeks longer, we're getting more biomass again. But for the most part, that kind of followed what we would expect. And in 2024, you can see, 5, 6, 7, 10, 20 times as much biomass that second year than the first. And so there are a lot of really neat interactions between how much space was available for the rye to expand. If you look at the 30-inch rows, the late termination timing had the most biomass, which was really pretty unexpected, but a neat demonstration of what rye can do if it's given enough water and growing degree days and space. And from a researcher's perspective, this is really interesting because these kind of serve as boundary conditions. So 2024 is almost a worst case scenario in terms of rye biomass and corn production, whereas 2023 is probably about average. So Rochelle, can you share how these crazy biomass numbers affected our seedling disease?
Right. So we went and sampled corn seedlings between V2 and V4 growth stage to look at seedling disease. And what we found was that in the drilled, where there was not that much distance between the corn and the rye, that had the most severe root rot compared to the other treatments. And the other thing that we can see here is that in 2024, possibly the interaction is that because we had more biomass, we possibly then that's the reason we had more seedling disease. So you can see that in 2024, there's more seedling disease than there was found in 2023.
I think, and one of the things about is once again in that 30-inch rows, right? The amount of seedling we see is very similar to that no cover crop, right? I mean, even when we plant green, we're still not getting that much more seedling disease. So I think that's really exciting to see.
Yeah, so this is really a fascinating part of the study because if we move on to the next slide looking at corn yields, we see that despite some differences in seedling disease, in 2023, we don't see an effect of the rye cover crop on the corn yields. Right, and so this kind of goes back to the study that Rochelle was speaking about earlier, where some years that there's that relationship between seedling disease and corn yield, and some years there's not. And we have a very specific example and a pretty clear understanding of why there's not one in our year. But it'll be neat to continue to unravel that relationship. So again, in 2023, we're really not seeing any effect of the cover crop on corn yields, either between the different treatments or the control itself. But in 2024, we're seeing up to a half of the yield reduction in that drilled cover crop, the positive control, and even in that skip row with the late termination. So you can see that the yields were already fairly low in 2024 just from growing conditions at about 180 bushels per acre. The cover crop's cutting that in half with the late termination. So that, again, is kind of a farmer's worst nightmare and a good boundary condition of how much we can actually lose from having this extreme biomass production. But one kind of positive note that we do see is that some of our mitigation strategies are pretty effective, especially looking at those wider spaced rows. With early termination, it's 30 bushels an acre, but it's not anything like those other treatments we were seeing. And then again, for both of the spacings, planting green is causing more and more yield decline. But kind of the second part of the story is, yes, we're seeing what it's doing to corn yields, but how is that affecting the services that we want to get out of the rye? And again, one of the big ones in Iowa is erosion control and managing water, especially in the spring. And so one important thing that having rye out there will do is increase infiltration. And pretty interestingly, across all the cover crop treatments for both years, we saw that the cover crops increased infiltration compared to no cover crop control, but within the different treatments, we're really not getting much of a difference. And so adding any amount of rye out there is going to improve infiltration. And actually adding more doesn't necessarily result in much more infiltration at all. And then in combination with that is, if that water's not going into the soil and it's running off, how much soil is it taking with it? And this is where we did see a difference with the different years, where in 2023, following the same trends as the infiltration, if you have a cover crop out there, it's going to reduce the erosion pretty much up to by half. But again, the different cover crop treatments didn't really affect it. So having less cover crop out there for less of the time still reduced erosion as having the entire field planted, which was really a pretty surprising finding and promising for adding that kind of to our toolbox. But then in an atypical year of 2024, we're seeing that having that rye cover crop essentially eliminates erosion, especially in the fully planted field and even the skip row with the late termination. And so this kind of comes back to trade-offs with what our intent for the cover crop is. If erosion's a major concern for you, then getting control with the cover crop is very attainable. This really is demonstrating when you have more biomass, you just won't have any erosion at all, essentially. And so to kind of summarize that interaction, we're really seeing that our mitigation strategies with the planting green and social distancing is reducing the corn seedling disease. And it mitigated the yield drag slightly in 2024, whereas there wasn't a yield drag in 2023. And then in terms of infiltration, we were really seeing, again, the cover crop in any way, shape, or form is improving that, as well as erosion. So these were really exciting findings and starting to help us understand what kind of capabilities we can have moving forward.
So I wanted to talk about some other research that we've been doing, trying to understand why this yield drag is occurring occasionally after cereal rye. And so one of the factors that I brought up at the beginning, one of the factors that people have blamed for this yield drag is nitrogen immobilization. And so we've been looking at that a little bit. Our hypothesis is that the available nitrogen in the soil is tied up by the decomposing winter cereal rye, right? So winter cereal rye has a very high carbon nitrogen ratio. And so as the organisms break that down in the soil, they need to use any available nitrate to help them break down that cereal rye biomass. And so there's no nitrogen then that's available to the corn seedlings. And so perhaps those corn seedlings are more stressed, and because they're more stressed, they're therefore more susceptible to infection by seedling pathogens. So this is kind of our hypothesis. And we've been working with Mark Lick's lab and trying to test this hypothesis. And I'm going to pass this to you, Rochelle, and ask you to explain what we've been doing there.
Okay, so this project was looking at different rates of nitrogen, also having a cover crop versus not having a cover crop, and finally slope position. So we either planted or did not plant winter cereal rye. When we drilled those plots, then those plots were terminated at 3 days after planting the corn. And we had several different rates of nitrogen, 0, 60, 120, 180, and 300 pounds per acre. And those were applied at corn planting. And then we had 3 different slope positions, summit, back slope, and toe slope. And then what we did is we sampled corn seedlings at V2 to V4 corn growth stage to assess for seedling disease. And what we found was when we looked at if there was effect of winter cereal rye on radicle rot of the corn seedling root system, just looking at the radicle and different slopes, what we found was that the radical rot was more severe following the cover crop, following the cereal rye at all slope positions. There was a significant difference versus if there was winter cereal rye or if there was not. And then we also found that radical rot was more severe in 2022 versus the other 2 years. And so we looked at that effect, and then we also looked at the effect of nitrogen when we applied it at planting, and the effect then on the radical rot for the 3 different slopes. And we found that the data did not support our hypothesis, which was that because the available nitrogen was being tied up by the decomposing cereal rye, that would then stress our corn seedlings and therefore have an increased risk of seedling disease. So our data did not support that hypothesis. But what we did find, though, was radical rot was more severe with nitrogen at planting, and then also that radical rot increased as we increased the pounds per acre of nitrogen. And so that was quite interesting, and I don't think we were expecting that, but it happens, and it tells us something.
So the other thing that we've been doing in my lab is looking at allelopathy and seeing if allelopathy is responsible for this corn yield drag. And this has been a challenge, but we've been making some small steps. And I have a student working with me at the moment on his PhD, so he'll be looking at this. But our hypothesis here is that the allelochemicals that are produced by the winter cereal rye are phytotoxic to the corn. And because of that, the corn seedlings are stressed as they're trying to grow, and so therefore they're more susceptible to infection by seedling pathogens. And we started this work actually in the lab there was a scientist working in my lab, Jyotsna Acharya, and she started this work where she grew rye seedlings in pots. She flushed out the exudates from those rye seedlings, put those rye seedling exudates into a petri dish, and then in that petri dish we grew Pythium, which is the main soil-borne organism that causes the seedling disease on corn following cereal rye. And so we had the allelochemicals, the Pythium, and the corn, and what we saw is we had more severe corn rot than if we just had Pythium alone. And so what Esteban is doing for his PhD is he is looking at the effect of winter cereal rye variety on seedling disease. And so we know from the older literature that different varieties of rye produce different amounts of allelochemicals. And we also know with some work that was done by Tom Casper a couple of decades ago that different varieties can cause different amounts of yield drag on corn. And so what we wanted to look at was see if that was related to seedling disease and also to yield drag. So this is a figure from one of Esteban's posters, but just showing that we have these 4 varieties of cereal rye, and with our no cover crop control, we plant corn into the cereal rye, the no cover crop control, look at seedling disease at around about growth stage V3, and then we harvest the corn at the end of the growing season to obtain yields. And 2024 was the first year that Esteban did this research. So if we look at seedling disease, first of all, that we can see that we had more severe seedling disease following a cereal rye cover crop, and that seedling disease severity varied among the varieties. And so we had more severe seedling disease following Serafino than we did following Aristuca or Ryman. So that was really interesting, fun to see this, well, fun from a pathologist's point of view to see this in the field. When we looked at the yields, The yields were reduced following the winter rye, but we didn't see a difference among the varieties. So this trial will be repeated in 2025 and hopefully in 2026, just because 2024 was such an anomaly of a year. You saw that in the work that we did with Peter, that we had so much biomass, and the case with this trial as well, we had cereal rye that was 6 foot tall. So, and then, of course, we're interested in the allelopachemicals. And so we've been working with Dr. Silvina Arias in the Seed Science Center, and she's been helping us develop and optimize a method for detecting those allelopachemicals. And so that's what we're in the process of doing now. We collected soil samples from the field when the corn was planted and soon after the corn was planted. And so we'll be looking at the allelopachemical quantities and hopefully finding something in that data. So just finishing up this episode, based on the data that we've shown you, our recommendations for corn following a winter cereal rye cover crop are to wait at least 10 days to plant the corn after terminating that cover crop. And the reason for that would be to just wait for those seedling pathogens to go down. That would be one reason that it's helping mitigate yield drag. There's also lower amounts of biomass that could also allow allelopachemicals to decrease after the rye has terminated. And there was one other thing I was thinking about. Oh, when we terminate that winter cereal rye earlier, then it's younger, and so its carbon to nitrogen ratio is lower, right? So that can help. But from the research that we've done with Peter, combining that planting green and then social distancing, so separating that corn or the rye from the corn, that looks like that it might be a worthwhile practice to help mitigate that yield. So we have several people to acknowledge for this work. Ben and Gavin work with you, Peter, right? So they're managing the trials, and yeah. And then John works with me. Fernando works with Mark Lick, so managing trials there. Esteban, Mila, and Ali are graduate students who work in my lab and Mark Lick's lab, and who have been helping us with the seedling disease stuff. We've also had numerous undergraduates that work with us. Thanks to Mark and Marshall and Silvina for collaborating with us. And a big thank you to members of the Precision Sustainable Agriculture Common Experiment 2 team who helped us with that planting green study. And of course, we have to acknowledge our funding, which came from Iowa Nutrient Research Center for a lot of our work here in Iowa, and then the USDA NIFA for funding the Precision Sustainable Agriculture project. And I'd like to thank you for being here today and for supporting Crops TV. If you have any questions or comments about this presentation, please feel free to reach out to me. And if I can't answer your question, I will be sure to pass it on to Rochelle or Peter, and I'm sure they'll be able to answer the question. But thank you for joining us today. And I want to thank Rochelle and Peter for joining me and making this episode a little bit more interesting than just one person. So thank you.